Hi, my name is Paul Cullen. I'm at the University at Buffalo in the Department of Biological Sciences. Thanks a lot to Lab Roots for allowing me to share our work with you today. It seems like an exciting event to talk about these different aspects of cell biology. The work I'm going to tell you about today comes from my laboratory at the State University of New York at Buffalo, or UB. And I'm excited to jump in and tell you about our research. Uh, our research generally has to do with signal transduction pathways. And in particular, we're interested in how signal transduction pathways regulate a process known as cell differentiation. Stem cells and other cell types can specialize into particular shapes and patterns where cells have unique functions. During this process, signal transduction pathways play a critical role. One type of signal transduction pathway are called MAP kinase pathways. And shown at the left of this slide here is a generic MAP kinase pathway that was taken from a textbook. And what you can see is how a signal, shown in orange here, is detected by some type of receptor. And a cascade of relay molecules transmits a signal, which ultimately results in changes in gene expression in the nucleus that allows the cell to respond to that stimulus. One reason why signal transduction pathways are important to study is because they play an important role in human health. And the misregulation of signaling pathways, in particular MAP kinase pathways and other pathways, can lead to diseases such as cancer in humans. So if we understand how signal transduction pathways function, this will tell us about how to help cure human diseases like cancer and other diseases. Unfortunately, signal transduction pathways are very complicated. And in this picture I've taken from a review article, we can see that unlike the simple model that I've shown you, signaling pathways are actually composed of many different types of proteins, shown as circles here, that are connected to each other in complex ways, which can be seen by the lines between the circles. And so how signals flow through complex networks to create specific responses is an important challenge in understanding signal transduction. To meet this challenge, we use a simple model system. And the logic of using model systems is that they can be simpler than the systems that are occurring in human cells. And in addition, there are particular specific tools that can be used that are available in many of these organisms, particularly genetic tools. And in this analogy here, I'm using a carburetor from a lawnmower. This left is the carburetor for a lawnmower, which can be used to understand how carburetors work before attacking more complex engines, such as the carburetor shown for a Cadillac engine, which is more complicated and perhaps harder to understand how it works. So if we use a simple model system, like some of the organisms shown in this picture here, this can help us to understand generally how regulatory pathways work in organisms. And because of evolution, the same types of pathways and processes are conserved between organisms. So if we learn something in yeast or zebrafish or fruit flies, this can apply generally to many different organisms. The system that we study is a fungal system, and fungal species are exist in many different contexts in the world. Mushrooms are fungal species, uh, and there are wild fungal species. There are also fungal species that interact with other organisms. So, for example, Candida albicans, shown here, is a commensal organism, but it can also lead to disease in humans because of fungal pathogenesis, especially when our immune systems become compromised. In addition, another fungal species called Fusarium is a plant fungal pathogen. So studying fungal species can help us understand how biological systems are regulated, and it can contribute to our understanding of human disease. We're interested in a process called filamentous growth, which is the ability of fungal species to change their shape and form elongated filamentous tube-like structures, as you can see for Candida albicans and Fusarium. And this allows these organisms to go into new environments and obtain nutrients. The specific organism that we study is the budding yeast. It's called Saccharomyces cerevisiae. And you can see at left, Fleischmann's active dry yeast is available in most supermarkets. And this yeast is used for us to bake bread, to make wine and beer. And it's a common organism that we use for food and industrial purposes. But we also can study yeast in the laboratory because it's a model genetic system. And we have powerful genetic tools to study processes like signal transduction and cell biology. In the micrograph at the top left, you can see the budding yeast growing in a form called budding. And yeast is a microorganism and a single-celled organism. Fungal species are eukaryotes, which are similar to plant and animal eukaryotes. And this budding yeast, when conditions are unfavorable for growth, can change into this filamentous growth mode. So our laboratory is interested in understanding how yeast cells differentiate into the filamentous growth pattern as a model to understand eukaryotic cell differentiation. In particular, we're interested in the cues that trigger filamentous growth and in the signal transduction pathways that are required to allow these cells to change from a yeast formed cell to a filamentous formed cell. And so what I'm going to tell you about today are three topics that we've made progress on in our lab that have to do with the regulation of filamentous growth. And the first topic I'm going to tell you about today is a type of protein called signaling mucins and how these proteins regulate MAP kinase pathways. So each of these stories I'm going to tell today will hopefully help provide some insight into how filamentous growth is regulated by yeast. And this may provide general insight into how signal transduction pathways regulate cellular processes like cell differentiation in eukaryotes. One of the first experiments that I did studying filamentous growth in yeast came from the discovery that when you starve cells for glucose, which is, of course, a common nutrient for many organisms, including us, yeast cells will switch their growth pattern and undergo filamentous growth. So we knew that when cells are sensing glucose limitation or glucose depletion, this must activate pathways that regulate the filamentous growth response. To identify what proteins might sense this glucose limitation, we performed an experiment called an expression profiling experiment. This experiment was done a long time ago using DNA microarrays, but more recently, people use comparative RNA sequencing analysis to compare the expression patterns of genes between two conditions. We found that some genes are induced when cells are limited for glucose, and one of the genes that we identified was a gene called MSB2, which encodes a protein that is a signaling mucin in yeast. This is a target of the signal transduction pathway that we study, but it also is a component of that pathway. So sometimes signal transduction pathways induce the expression of genes that encode components of pathways to create a positive feedback loop. This protein, called MSB2, is a member of the signaling mucin family of proteins, which are highly conserved among eukaryotes, and they're found in many organisms, including humans. Signaling mucins have certain properties, and you can see MSB2 shown here as a transmembrane protein that has an extracellular domain that is highly glycosylated, a transmembrane domain that passes through the membrane, and a cytosolic domain that interacts with cytosolic proteins to regulate signal transduction pathways. On the extracellular side of MSB2 are tandem repeats that are rich in serine, threonine, and proline residues. And you can see that MSB2 has about 7 of these repeats. So the fact that we identified a signaling mucin in yeast allowed us to do genetic experiments to understand how signaling mucins regulate MAP kinase pathways. And the MAP kinase pathway that we study, we call it the F MAP kinase pathway, which stands for filamentous growth. So we used yeast as a model to understand signaling mucins, because signaling mucins are important in regulating MAP kinase pathways in humans and other species. And signaling mucins are also important in human health. In fact, in many cancers, signaling mucins are upregulated, and they provide a diagnostic tool for cancer diagnosis. And in fact, immunotherapies target mucins to try to treat cancers in humans. Nevertheless, how signaling mucins function at the time we began these studies was poorly understood. So I want to show you a couple of experiments here that helped us to understand how mucins function. And the first experiment you can see on the left here, we introduced an epitope tag into the extracellular domain of MSB2, and we found that part of the protein is released from cells. And you can see in the model here, there's an arrow which shows that the glycosylated part of MSB2 is released from cells, and it floats off into the media and can be detected in supernatants. So here, P stands for pellet at left, and S stands for supernatant. So this means that MSB2 is cut, and we identified a protease called YPS1 that cuts MSB2 and releases the extracellular domain of the protein. By comparison, a cytosolic tagged version is only found in pellets in the middle immunoblot that I'm showing. We also found that the extracellular domain of MSB2 has an inhibitory function. And you can see by these fuse1-laxi beta-galactosidase assays that, and I'm not sure if you can see this, but cells lacking the mucin domain of MSB2 have actually higher levels of MAP kinase pathway activity. And this reporter gene is an indicator of the activity of the MAP kinase pathway. And we can get rid of large portions of the extracellular domain of MSB2, including from amino acids 100 to 818, 900, 950, and we can create a hyperactive version of this protein. Therefore, in the model, the active form of MSB2 is this much smaller protein that lacks the large extracellular domain. This discovery led to a cleavage-dependent activation mechanism for signaling mucins, where the cleavage and release of MSB2 leads to the activation of the protein and the activation of the MAP kinase pathway. But how does this connect to glucose limitation? How does glucose starvation activate this type of sensor protein? And the answer to that question came from an additional study that connects to another pathway called the unfolded protein response, or UPR. We found that the unfolded protein response, which is a signaling pathway that operates in the endoplasmic reticulum of cells, is required for the activation of the pathway. And you can see this here. Here's a protein called IRE1 in the endoplasmic reticulum, which detects unfolded proteins. And when unfolded or misfolded proteins are detected in the endoplasmic reticulum, this allows IRE1 to become activated and induce the expression of genes. And one of those genes encodes the protease that cuts MSB2. In the immunoblot below this model, you can see that cells lacking IRE1 are defective for the activation of the MAP kinase pathway based on an approach called a phosphoimmunoblot analysis, where the phosphorylation of a protein, here KIS1, which is the MAP kinase for the MAP kinase pathway, fails to be induced when cells are grown in glucose-limiting conditions using a poor carbon source called galactose, or GAL. And so this led us to a model at right where the glycosylation of MSB2 becomes reduced under glucose-limiting conditions. And the reason for this is because the glycosylation of proteins involves a sugar or oligosaccharide modification. So when carbon source limitation occurs, the oligosaccharides required to glycosylate proteins becomes reduced, and this allows MSB2 to be recognized by the unfolded protein response as a misfolded or unfolded protein. This quality control UPR pathway induces the expression of YPS1, which is the protease that cuts and causes release of the inhibitory domain of MSB2, therefore activating the protein, which leads to high MAP kinase pathway activation. In this way, MSB2 functions as a glucose sensor, where when glucose levels are high, MSB2 is fully glycosylated and not active. And when glucose levels become limiting, MSB2 becomes underglycosylated and is then activated. So in this way, we think that signaling mucins can function to regulate signal transduction pathways by the state of glycosylation of their extracellular domains. So that was the first story that I wanted to tell you today. The second story that I'd like to tell you about relates to the first story, and it has to do with the concept of signaling specificity and how scaffolds affect signaling specificity. And in order to tell you this story, I'd like to introduce to you a puzzle. And here's the puzzle. In the center of this slide, you can see the MAP kinase pathway that regulates filamentous growth. At the top, you can see nutrient limitation, which activates the signaling mucin MSB2, which sets off a cascade of events where signaling proteins lead to the activation of a MAP kinase cascade. You can see the MAP kinase KIS1 in red at the bottom of this pathway, and KIS1 activates the activity of transcription factors that induce target genes that lead to filamentous growth. In yeast, this MAP kinase pathway shares components with two other MAP kinase pathways. A pathway shown in blue at the left, which allows yeast cells to undergo mating, and a green pathway at the right, which allows yeast to respond to osmotic stress, called the hog pathway. All of the proteins shown in black are shared between more than one pathway, And in fact, the proteins that are shown in the gray box are required for the activation of all three pathways. So how does a cell, when a signaling pathway gets activated, not activate all three pathways? And the answer to this question comes from the function of a type of protein called scaffolds. Scaffolds direct general components to a particular pathway. We can see that the mating pathway employs a scaffold called sterol5, which binds to general factors and pathway-specific factors to promote a pathway-specific response. Similarly, the hog pathway is activated by a scaffold called PBS2. We identified a scaffold for the filamentous growth pathway called BEM4. BEM4 is shown as this large blue protein which binds to a type of protein called a Rho GTPase. This Rho GTPase is called CDC42 and is evolutionarily conserved in most eukaryotes. CDC42 can be activated by binding to GTP, and CDC42 in its active form turns on the MAP kinase pathway. In fact, CDC42 is a general component that functions in all three MAP kinase pathways. The identification of BEM4 in our laboratory, which is a type of protein called a RAL-GDS protein, at least BEM4 has homology to these proteins, its interaction with CDC42 leads to a pathway-specific response in that CDC42 only regulates the filamentous growth pathway in a BEM4-dependent manner. So how does BEM4 direct CDC42 to a particular pathway over other pathways in which that protein might function? And the answer to this question came from looking at the levels of CDC42 protein in the cell. In this immunoblot, you can see with antibodies against CDC42 that the levels of the protein are lower in cells lacking BEM4. And this BEM4 triangle refers to cells that don't contain the BEM4 protein. So this told us that BEM4 may stabilize the CDC42 protein. One way that proteins become destabilized is by their turnover by a ubiquitin-dependent manner, where proteins are modified by post-translational modification by ubiquitin, which leads to their turnover in the cell. And at this immunoblot on the right, we can see that cells lacking BEM4, shown by this white circle, show higher levels of ubiquination of CDC42, as shown by this smear by co-IP ubiquitin that is detected when CDC42 is immunoprecipitated from the cell. Therefore, BEM4 may protect CDC42 by preventing its ubiquination. And this may lead to higher levels of CDC42, which then leads to the activation of a MAP kinase pathway. We drill down a little deeper into this question by generating versions of CDC42 that are active and that cannot be turned over. And if you look at the immunoblot on the right here, you can see that the phospho-KIS1, shown in red, occurs at higher levels in cells with an activated version of CDC42 called Q61L. In fact, the mating pathway, here shown in green, is also at higher levels in cells containing Q61L, and that can be seen by the phosphorylation of the MAP kinase pathway for the mating pathway called Fuse3. However, cells that fail to turn over active CDC42, shown by CDC42 Q61L plus TD, show higher levels of phospho-KIS1 compared to phosphofuse3. Therefore, when there's more CDC42 in the cell, presumably because of protection by BEM4, you get more activation of one MAP kinase pathway, the red pathway or the filamentous growth pathway, versus another MAP kinase pathway, or the green pathway. Putting this all together, we have a model where CDC42 gets activated by the mucin MSB2, and this can then lead to the turnover MSB2 by the polyubiquination of the protein and its destruction in a protein component called the proteasome. This is shown by the green arrow where CDC42 is turned over and degraded to turn off MAP kinase pathway activity. Scaffolds, like the scaffold BEM4, can act against this turnover mechanism to protect CDC42 from turnover, which leads to higher levels of filamentous growth pathway activity. And this shows how scaffolds can bias signaling by protecting Rho GTPases and potentially other proteins from turnover. Presumably, there are other specificity mechanisms that remain to be discovered in yeast and other organisms that share components, that have pathways that share components. So in the final project I want to tell you about today, I just want to mention how signal transduction pathways can function not as individual entities, but in networks where multiple pathways coordinate signaling with each other. I've told you about how a MAP kinase pathway can regulate filamentous growth, but in fact, multiple pathways are required for filamentous growth in yeast. And shown here are 5 or 6 different pathways that regulate this response. We performed an experiment to look at the secretion of MSB2 in a panel of mutants where every non-essential yeast gene was deleted in an ordered collection. And if you look at this panel A and this slot A2, you can see that MSB2 secretion is reduced in that particular mutant. And in this experiment, we identified genes that impact the secretion of MSB2, which led to the identification of new regulators of MAP kinase pathway activity. This actually was a big experiment where we looked at every gene, non-essential gene in the genome, which is almost 5,000 mutants that was done in duplicate. And what this showed us was that many pathways in the cell regulate filamentous growth by regulating the MAP kinase pathway. And so you can see these pathways here, for example, RAS, OPI1, RPD3, regulate filamentous growth by affecting the activity of the MAP kinase pathway. Some of these results verify previous studies connecting some of these pathways together. For example, RAS has been previously connected to regulating the MAP kinase pathway by work by Jerry Fink at MIT. By expanding on this network, we showed that many of these pathways regulate each other's activities and target genes. And shown at right, we can see pathways shown as circles in different colors that regulate each other's target genes by arrows connecting one pathway to another. In a recent experiment by a graduate student in my laboratory, we explored the connection between these pathways under different conditions. In panel D of this paper by Vandermeulen and Cullen, we can see that different mutants show defects in filamentous growth that changes depending upon the condition studied. And each one of these words up here on the x-axis, YPD, YEPCAL, are different conditions. And what this experiment showed us was that different pathways in the network play different roles under different conditions. And so we have pathways that play a more important role are shown as bigger circles, and pathways that play less of a role are shown as smaller circles. Therefore, the network that regulates a differentiation response can change in a regulated manner. So in conclusion, today I've shown you that signaling mucins regulate MAP kinase pathways and revealed a cleavage-dependent activation mechanism that applies to signaling mucins. We've shown that signal specificity can be regulated by scaffolds, and scaffolds can protect G proteins from turnover to bias signaling from one pathway over another. Signal transduction pathways, like MAP kinase pathways, function in complex networks, and these signaling networks can change based on the environment. We have a lot to learn about how these signaling networks work and how signals are coordinated to create integrated responses. So I feel that our work is just getting started. All of the work that I've shown you today comes from the members of my laboratory, and this is just a snapshot of just one generation of laboratory members who've worked on these projects. And this was actually turned out to be quite a party a couple of years ago. So I would like to formally acknowledge the current and former members of my laboratory, many of which have contributed to the work I've showed you today, and some of which contributed to projects that I didn't have time to tell you about. And we've also collaborated with many members of the signaling community. And I'm really proud to be part of a community that so freely shares information and reagents. In addition, our work has been funded by the American Cancer Society and the American Heart Association and by the National Institutes of Health. So this work is funded by these agencies because of evolutionary conservation. What we learn by studying signaling pathways in a simple model may apply to other systems, provide a general knowledge to understand signaling pathways, cell differentiation, particularly as it may pertain to human health. So with that, I'd like to take any questions. Thank you very much for your time. And again, thanks to LabRoots for this opportunity.